Thank you all for logging in to listen to my talk. My name is Luke Pearson. I'm a PhD student at the University of Southern Mississippi, studying the conservation genetics of the alligator snapping turtle here in the state. So as many of you know, the alligator snapping turtle is a large freshwater turtle that's mostly aquatic, and it's found in the southeastern United States. This species was historically harvested for the turtle soup industry, which caused significant population declines due to the removal of reproductively active adults. The removal of these reproductively active adults caused significant declines in recruitment into the population. So once these population declines were noticed, commercial harvest was banned in all states, and currently recreational harvest is only allowed in Mississippi and Louisiana. There is an ongoing species status assessment to determine if Macrochelys, both temminckii and swaniensis, are warranted federal protection under the U.S. Endangered Species Act. And for a species status assessment to be valid and accurate, we need current, up-to-date information on distributions, population estimates, and conservation genetics and the genetic assessments of these populations. So currently there are two range-wide genetic studies on Macrochelys temminckii. The first of which was by Roman et al. In 1999, which used mitochondrial DNA to determine that there was little to no movement of females across river drainages. So it seems that there are unique haplotypes in almost every river drainage that they sampled. So 8 of the 11 mitochondrial haplotypes were unique to single river drainages. And this study was further supported by Kelly in 2014, who used microsatellite loci, which also determined that there is little to no movement of males or females between these river drainages. They also determined that there are 6 evolutionarily significant units, one of which includes the Pascagoula River here in Mississippi. Both of these genetic studies shone a light on the Swanee population of alligator snapping turtles, which in 2014 was currently determined as its own separate species. So what's interesting about both of these two genetic studies is that only 13 samples were used from the entire state of Mississippi, which is in the heart, the core of this species distribution. And all 13 of these samples came from the Pascagoula River. That means that there was no samples taken from the Pearl River, the Upper Tombigbee River within the Mobile River drainage, the Big Black River, and the Yazoo Rivers, which are both part of the Mississippi River drainage here in western Mississippi. So to kind of fill in this gap, this lack of information for Mississippi, we had two main objectives. Does population structure exist among the major river drainages in Mississippi, similar to the pattern found by Roman and Kelly? And if it does, if there is population structure, does this population structure also exist within a single river drainage? So is there population structuring within the Pascagoula River or within the Pearl River? For the first objective, we do expect to see the same pattern that was found by Roman and Kelly. We expect to see these strict, discrete populations in each of the river drainages with very little to no migration between them. And this makes perfect sense. Alligator snapping turtles, although they do move over land, these movements are infrequent and they're usually for very short distances, which completely limits migration between river drainages. However, two separate things that could occur with population structuring existing within a river drainage. We know that temminckii are long-lived, and we know that they can move long distances when they want to, upwards of 20 kilometers within a couple years. So this long-lived nature and the potential to move long distances increases the potential for genetic dispersal within the river system, and this could lead to little or no population structuring. However, on the flip side, temminckii tend to inhabit core areas of their home range most of the time. They don't move out of these areas frequently, and when they do, they usually come back in a relatively short period of time. So this site fidelity may actually create significant population structuring, even within fully connected systems. And this is kind of seen by Tier in 2010, where she found discrete populations in population structuring in a Georgia population versus a Florida population of alligator snapping turtles. Currently, I have approximately 600 samples from alligator snapping turtles from 56 sites across the state of Mississippi. Currently, I've genotyped 246 of these samples from Mississippi and 28 samples that were donated to me from Tennessee by Josh Ennin. Most of the genotype samples come from the Pascagoula River drainage, shown here in red, in southeast Mississippi. However, I have 56 samples genotyped from the Pearl River drainage here in purple, and 52 samples from the Big Black River drainage, which is part of the larger Mississippi River drainage in western Mississippi. All of these samples were extracted using a DMEZ kit and genotyped using established microsatellite loci from Hackler, as well as new loci that I developed for temminckii. So when we break it down and we look at the pairwise FST values, as well as the structure analysis, what we're seeing is quite significant population structuring and genetic isolation between each of these river drainages. So we're seeing a range of 0.17 pairwise FST between the Big Black and then the turtles from the state of Tennessee, all the way up to 0.34 between the Pearl and the state of Tennessee. So we're seeing very significant isolation between populations and genetic structuring between river drainages. And this is further supported by the structure plot. So each bar within this plot represents the ancestry of an individual turtle. So as you can see, the Pascagoula River turtles are very strictly purple. That means that they have extremely high levels of ancestry for the Pascagoula River. We see the Pearl River separated in red, Big Black River separated in green, and then the Tennessee State turtles here in blue. The Tennessee State turtles, at least the last few turtles that you can see on this structure plot, do show some admixture with the Big Black River, and this is assumed to be because those turtles are not actually native to Tennessee and were reintroduced from populations in Louisiana, which kind of explains why they are more admixed ancestry. What we're also seeing is one individual within the Pearl River that has a very high level of ancestry for the Big Black River. As you can see, this picture here, this is a 50 centimeter carapace length male, approximately 30 kilograms. The interesting part about this male is that he had 4 supermarginals on both sides. 4 supermarginals is exceedingly rare to find in the Pearl River, as 95% of alligator snapping turtles within the Pearl River drainage only have 3 supermarginals. So this individual had 4 supermarginals, and that is extremely common in the Big Black River. So it's likely that this turtle was captured on a trot line or a limb line, taken home by somebody to show their family, their friends, show off this large turtle that they caught, and then they got lazy, they didn't want to take it back to where they caught it, and they released it in the closest body of water, which was the Pearl River. So this is kind of a human-mediated translocation, and if I managed to catch a turtle that was translocated from the Big Black to the Pearl River, then there's a chance that this translocation is also happening at a much higher frequency than what we were originally thinking. So we know that we are seeing strict, discrete populations within each of these river drainages, which makes sense. Now we're going to look at the within drainage population structuring of the Pascagoula River, which is the largest undammed river system in the contiguous United States. And what we're seeing is three genetic groups. We're seeing Upper Leaf River, a group that includes the Upper Leaf River, shown in green, a group that includes the Upper Chickasahee River, shown in red, and then we're seeing a Pascagoula River group, shown in blue. So there is admixture between these populations. As we can see on the last columns of the Leaf River group, we are seeing a lot of admixture between the Pascagoula River and the Leaf River, and that's because each of these groups, you're seeing an upstream to downstream effect. So you're seeing admixture the further downstream that we get to the Chickasahee River and the Pascagoula River system. And this is really well by many other freshwater turtle species. So the structure plot on the top is my temminckii. The next one is the Pascagoula map turtles found within the Pascagoula River drainage that shows the same thing. You see a Leaf River group, you see Chickasahee, Upper Chickasahee River group, and then you see a Pascagoula River group. It's also shown in the yellow blotch sawback, Graptomys flavomaculata, Upper Leaf, Upper Chick, and then Lower Chickasahee and Pascagoula group, as well as razorback musk turtles, where you see red and black, which is part of the Pascagoula River, the buoy and leaf group, and then a Chickasahee group. So throughout the Pascagoula River drainage and throughout many different species of freshwater turtles, we are seeing this same pattern of three genetically discrete populations within this largest undammed and continuous river drainage in southeastern Mississippi. So in conclusion, we're showing the same type of pattern that Roman and Kelly found, discrete populations in each river drainage, and this kind of leads to the idea that if any extirpation occurs in any of these river drainages, that would create a loss of genetic diversity for the species as a whole. We're also seeing potentially human-mediated translocation, and it's probably occurring more frequently than we believe because of the fact that I managed to catch one of these translocated turtles. And finally, we are seeing intradrainage population structuring, especially within the Pascagoula River. We're having these three groups, the Upper Leaf, Upper Chickasahee, and Pascagoula River groups. And this is likely caused by historic sea level rises. So when the sea level rose, it created refugia within the headwaters of the Leaf River and the headwaters of the Chickasahee River, which created two genetically discrete populations. And that's what created the Leaf and the Chickasahee groups. And when these two rivers confluence, that would create the Pascagoula River group. And with that, I would just like to thank all my advisors, collaborators, and funding sources, MDWFP, Fish and Wildlife Service, Nat Geo, ATO. Thank you all for listening to my talk. And if you have any questions, please feel free to email me at luke.pearson@usm.edu. Thank you very much.